Thank you very much, Rayanne, and thank you, Melanie, for your introduction. That reminds me of all the good times we had, and it's always fun to see you continuing on with your career. So if you want to go to the first couple slides, Rayanne. Yeah, okay, next one. Yeah, just a couple more things about my background. I grew up in Wahpeton, North Dakota, and I worked for the Indian Health Service from 77 to 98, and then for another 10 years in a private clinic in Dunseith. Seen most of the same patients I'd seen before, but in a private setting. And then began with the Strong Heart Study in 98 and teaching at genetics at Turtle Mountain in 1998 as well. Continuing on today with our research projects there. Next slide. Next slide. Yeah. So I won't belabor this, but I think it is important to get a sense of the history of where we are with genetics. And many of you probably know that it was background. Oh, the slide before that, I guess. Seems to be one missing. Go ahead, back forward again. All right, okay, stop there. So Gregor Mendel made his first discoveries around the time of the American Civil War. And unfortunately, his discoveries were not appreciated for nearly 40 years until about the 1900s. And in 1908, Dr. Garrett in England first noticed that the same principles that Mendel had discovered in the mid 1800s were also at work in the inheritance of certain medical conditions, inborn errors in metabolism. And a couple of decades later, a condition called PKU, or phenylketonuria, was discovered, where certain individuals had changes in their pigmentation. They developed progressive intellectual impairment, and these children were born in families that recapitulated Mendel's rules of inheritance. So it showed that humans were also susceptible to these same rules that he developed in pea plants. And we'll talk a little bit more about that later. The amazing thing is that just a couple decades later, the actual enzyme that causes PKU, the deficiency of that enzyme that causes it, was discovered. And very shortly after that, within a few years, trials were began to use a dietary treatment to improve life for these children. And then a simple screening test was developed, which now is used essentially universally throughout the developed world. So next slide, please. We won't dwell here, but I was fortunate to be able to visit the monastery where Mendel did his work in the Czech Republic a few years ago. And this is me in the garden they have memorializing his work. It may look like I'm fertilizing his pea plants, but I'm actually just fondling them, and enjoyed being there in that particular place. Next slide. So one of the first applications of genetic understanding was to begin to learn about the blood types that people have. And this was very useful in allowing for safe transfusion of blood. And in 1937, a new blood type was discovered, the Rh factor. And this improved transfusion safety as well. But in addition to that, it allowed us to understand what was happening in a particular serious condition of newborns called erythroblastosis fetalis. In this situation, the mother is an Rh negative individual, and she conceives a child who's Rh positive. And unfortunately, her immune system recognizes this child as foreign and works against it, causing severe anemia, which can eventually kill the child in utero or cause serious problems even after birth. So in 1968, a treatment was developed called RhoGAM, which is essentially a type of vaccination which keeps the mother from being sensitized to the Rh positive infants. And so the children, like you see in the upper left there, that have severe jaundice when they're born, we essentially don't see this problem at all. It's essentially been eliminated completely from medical experience. You won't find medical students today will probably never see a child affected this way. Children are still have problems with jaundice, but it's from a different cause and not from this particular cause. So it's an amazing story of essentially eliminating a condition that used to claim the lives of many hundreds and hundreds of children in the past. So next slide. PKU, we've already discussed a fair amount. It is probably one of the most striking examples of being able to use the knowledge of a genetic condition to change the environment, in this case, the diet of the child, to prevent a very serious intellectual handicap. So some of the children you see here have been affected. They probably grew up in a country that was not doing screening. They didn't have the benefit of that change of diet as a young child, and they're permanently disabled as a result of that. Whereas children that are discovered in the newborn period through screening and maintain that diet, live perfectly normal lives and go on to be normal in every respect, except having to pay attention for their diet. Next slide. So those are interesting from the past, but what about recently? So in 2003, it was discovered in some French Canadian families that there were individuals that had mutations or variations in a particular gene called PCSK9. And this increased the level of the bad cholesterol, the LDL cholesterol that they have. And this was confirmed in another study. But they also noticed in the Dallas Heart Study a few years later that there are other variations that actually decreased the bad cholesterol, not increased it. And this is a good example of sometimes different mutations in the exact same gene can have opposite effects. They can either improve or cause problems for your health. The Strong Heart Study has also found these similar variations in the Indian population that participated in our study. And it was noticed in the ERIC study in 2006 that individuals who had inherited these beneficial mutations from both of their parents actually had 99% drop in their risk of heart disease, really dramatic change. And a few years later, in 2015, two drugs were developed, which were based on the effects of this PCSK9 gene that lower the bad cholesterol for people that have had this problem. And this has been a new treatment that has augmented the statins that so many people use to reduce their blood cholesterol. And it's been a welcome improvement. So next slide. So another drug that's very frequently used called 5-FU. This was discovered in the 50s, was one of the first drugs used to treat leukemia in children. And today it's used very widely for chemotherapy yet. I've actually taken it myself topically for little skin lesions I've had on my face, as many other people have done. But unfortunately, if you take it internally, about 3% of the population will have serious side effects. And in fact, some of them will die of this severe toxicity. And we've come to understand that there are certain genetic changes in the DPYD gene that causes this problem. And nowadays, standard of care is that if you're going to be started on this medication, they will do genetic testing to see if you happen to carry any of these genetic changes that could cause serious complications for you. Next slide. Another drug which illustrates an interesting problem. This is in the area of pharmacogenetics, which is the interaction between your genetic makeup and medications that you might be taking, whether they might harm you in some way or whether they might be additionally beneficial. So this particular medication called Plavix, many of you might be taking it. It's used to decrease the clotting, especially for people that have had heart treatments such as stenting and so on. And it's an interesting medication in that it's not actually active in the pill, but once you get it inside your body modifies that medication and makes it an active drug. Unfortunately, some people carry genetic mutations that don't allow them to activate that medication. And so they can take it all they want. They don't get any benefit from it because it doesn't become active in their body. So we have found that about 30% of the population carry at least one of these variants or mutations that reduce the effectiveness of Plavix. And we know that it can, if they carry those kinds of modifications, it can increase their risk of clotting in their stents up to threefold. So unfortunately, this is not a particular form of testing that seems to have become standard so far, but I wouldn't be surprised if it doesn't in the near future. So next study or next slide. And we have done testing for these particular changes in this gene in the American Indian population, and we have found similar levels. So it's not something that simply applies to non-Indian people. Next slide. So where do we go from here? Well, there's lots of exciting things going on, and I'm sure you've heard sometimes alarmist news reports about genetically modified organisms and being able to change the DNA in people and so on. And whether we believe in that in terms of agriculture and so on is a different subject. But in terms of human medicine, there are many conditions that have very, very inhumane outcomes for children and for people. And if we could improve that by changing their DNA, a lot of people say, well, if there's something wrong with my gene, why don't we just change it? Well, we are coming to the point now where we can do that. And the technology that's being used now is called CRISPR, CRISPR, and it's now just beginning to be used in human patients. And here you see an individual who's one of the very first sickle cell patients treated with CRISPR to change the DNA that she's inherited to allow her to create a more typical normal hemoglobin and get around the serious problems that she has from her sickle cell disease. You might be aware that sickle cell is more common in people who come from populations where there's lots of malaria. And the interesting thing is that if you carry one of the sickle cell alleles from one of your parents, you're more resistant to malaria, and therefore it's been promoted in the population from an evolutionary standpoint. But if you're unlucky enough to carry two different alleles, then you have sickle cell anemia, which means that your life will be shortened. You'll have lots of very, very painful crises, and it's a very serious condition to have. So luckily, we're beginning to make progress and actually making changes in people's DNA when they've inherited conditions that are very serious, such as this. There's also a great deal of work being done in the area of cancer, not only to test the cancer itself to see what kind of mutations have occurred in that cancer, which allow us to guide the treatment to do a better job of treating the cancer, but also to better understand what caused it in the first place, what environmental factors might be in play, and so on. So, and also to create treatments where we encourage the immune system to fight the cancer itself. So, a direct treatment which is based on genetics as well. Next slide. So things have changed so rapidly, and I can't help but be enthusiastic about some of the technology we've seen. This is way beyond when Melanie was working with me in the lab, and we were doing gel electrophoresis to test for these genetic changes, and she did a good job. I am disappointed, of course, that she hasn't gone the route of genetics. She's gone more towards epidemiology, but we'll let that be for now. Maybe she'll still see the light and come back to us. At any rate, this in the upper left there is a device about half the size of a cell phone, and it's relatively cheap. We have one at Turtle Mountain Community College now, and you put a drop of solution in it, which contains the DNA you want to analyze, and it actually pulls the strand of DNA down through a tiny hole. And as it goes through that hole, it changes the electrical signals that are generated. and that allows you to determine what the sequence of the DNA is, the 4 base pairs, A's, T's, C's, and G's, and it reads them out for them, just like you're reading letters in an alphabet. And it does that at a very high rate and generates a tremendous amount of data. Right below that is another instrument we just recently purchased at Turtle Mountain, which also does sequencing, but it does it in a little bit slower and more focused way, but also somewhat more accurate way. So I put them up there just to illustrate that we are making progress and increasing the capacity in our tribal colleges, and teaching the students that we have working with us today for jobs that will be in the forefront in the future. So next slide. So you know a lot about the Strong Heart Study already, but I'd like to just emphasize that since 1988, the participants have been very generous, and they've been trusting, and I think we've earned their trust to care for the DNA that they have donated to the Strong Heart Study. And we continue to make discoveries and findings that come from that, and I think that is building important information for improving their health in the future. Next slide. So a couple of the studies that I've worked with, which I think illustrate the benefits of genetic research from a tribal perspective. The Preeclampsia Study is mentioned. Preeclampsia is also known as toxemia pregnancy in the past. And we've, thanks to Mel and others, we've recruited now a little over 800 individuals from Turtle Mountain, Cheyenne River, and Spirit Lake who've donated their DNA to help understand this. And we have found 3 small genetic changes in a particular gene that is associated with increased risk of preeclampsia. And this was actually the very first time that this was discovered anywhere in the world. And frankly, I was surprised that we made any novel contribution. It's just not that easy to do, but every once in a while people get lucky, and we did. And this has been verified and replicated in other populations, so it's not just in the Indian population that this occurs. Another thing you should know about preeclampsia is we used to think that once you were done with your pregnancy, everything went back normal and you were fine again. But we find that having had preeclampsia increases your risk of high blood pressure later in life, and also other cardiac disease. So it's important to understand more about it. Next slide. And this is Mel getting her PhD at the University of Minnesota a couple of years ago, and that was a really fun day. Next slide. So as Mel mentioned, we did engage the elders in our community, and I think that was a very important step. And here you see them getting together. Like Mel said, a number of them are gone now, unfortunately, but we really had fun. And we talked about a lot of other things besides genetics, and I think it was a learning experience all around. Next slide. And our students are really the main focus of our study. And we've had over 50 students now. A couple of them have gone on to become physicians. 2 of them are physician assistants. Many of them are in nursing, and over half of them have obtained their bachelor's degree and gone on to various places, some of whom are on the call. I think Jacob Davis has his master's degree now and has gone on to other work as well. So we've really been proud of our students. Next slide. Some of the young investigators that have worked with us have been more advanced in their training. Some of them, like Shailene Hall and Nevin Gillis, have worked with our study as students, but Candelaria was a Navajo who is practicing down in the Spirit Lake area and has helped us out there. Gilbert Falcon, Laramie Lundy, and Shailene and Nevin, as mentioned, are all physicians now, and have helped us at one time or another. Bethany Davis has a PhD in cell biology from UND, and is also joining us to do some analysis as well. So we've really tried hard to engage younger folks in this work. Next slide. So, and as mentioned, we've tried hard in our translational efforts between radio programs, YouTube videos, newsletters, and so on. Next slide. And next slide. The other study that's primarily focused on genetics, although not entirely, is the factors influencing asthma. And we had actually three different goals. One was to look at environmental factors that might influence asthma risk. Second, to look at genetic factors that would do the same thing. And thirdly, to see whether increased educational intensity would decrease the complications from asthma. But in terms of the genetics, we did find 10 different changes in the DNA that are similar to the non-Indian population, and that's been somewhat useful. And I think, again, we've done a lot of work to try to improve the care for asthma in general and our understanding of asthma in this way. Next slide. And we've been involved student researchers at Cheyenne River as well as up at Turtle Mountain. Most of the samples were analyzed at Turtle Mountain, but others such as Seth Bendigo and some from Cheyenne River here have also helped out. Next slide. And we've reached out beyond the Indian community. Dr. Dara Torgerson at the University of California, San Francisco has been very helpful. She came down to Eagle Butte and helped train some of our students down there and provided some additional funding, if you'll go to the next slide, which allowed us to study many, many more genetic changes. So at Turtle Mountain, we've looked at a little over 10 or 15 changes over time in these 300 and some individuals. But Dara was able to provide funding for us to use what's called a microarray or gene chip, which looks at over 2.5 million different genetic changes in each individual in each of these 300. So it generated a tremendous amount of data. And one of the interesting implications of this is that, as you know, in regular medicine, sometimes you discover things you didn't plan on. So you go to the doctor because you have a cough and they do a chest x-ray and he said, well, it turns out you don't have pneumonia, but by the way, you do have gallstones. So now you know something completely different that you had no plans to discover. And we're looking at 2.5 million different genetic changes. It's not that surprising that you would find one or two things that you didn't plan on. And in this particular case, we did find two individuals amongst these 300 that have a change in a particular gene that increases their risk of heart disease. And even though that's not having to do with asthma, we went back to them and explained to them what we'd found and asked them if they wanted to do further follow-up on this. And at least one of the individuals was in agreement to do that. And we did some further testing, confirming our results at the Mayo Clinic and doing the echocardiogram and so on to look at the wall of the heart wall and its thickness, because these individuals with these genetic changes tend to have an increased thickness in their heart wall, which sounds good in some ways, but it's actually a not a good thing to have, and they go on to have other complications of cardiac nature. Luckily, this individual does not show these changes at this time, and we've encouraged her to have routine testing every 5 years or so to make sure that they're not developing. But you might think that this is pretty standard. When we did the Strong Heart study, of course, if we found you had high cholesterol or high blood pressure, we would let you know right away. And we'd help you get an appointment in the clinic to make sure you got proper treatment for it. In genetic studies, this is really not done very often. And it may sound surprising, but most genetic research has not been doing this. It's becoming more common now, but partly because the genetic research was usually so far out in front, we didn't know for sure what we found was really going to be confirmed and was important. But in this particular case, these two individuals had the changes that we felt were clear cut, and they were important, and we went back to them. And I think it shows that not only in this research, but in the Strong Heart study, we're committed to informing people of whatever it is we find that can be useful to them. And I'm proud of this as an example of ways that we are, I think, continuing to advance research in general. The next slide. And the next slide. And just lastly, for a little fun, these are two snowmen. And one of them says to the other, I got one of those DNA kits the other day, and it turns out I'm 85% water, 8% dirt, and 7% dog pee. So if you're getting one of those genetic tests, you might want to just kind of take it with a grain of salt. Sometimes they're not quite as accurate as they claim to be. Next slide. And lastly, I just want to thank all of the hundreds of people who have trusted us and given us their time, their medical information, and their DNA. Thanks to the tribal governments that have been supportive and appreciative, Turtle Mountain, Cheyenne River, and Spirit Lake in particular. We really appreciate their support. The Indian Health Service has been very good and gracious in many ways, as Mel knows, in her work as well. And then, of course, funding from NIH and others. So that's all I have. I'm happy to take whatever questions you have in the last 10 minutes or so.